My name is Richard Anderson. I'm the James G. Boswell Professor of Neuroscience at the California Institute of Technology. I'm the principal investigator on this clinical study and also lead a group of students and postdocs who work on this project in my lab at Caltech. So at the imaging center at USC, we imaged the parts of the brain that were active in Eric when he imagined a movement. Since he's paralyzed, he can't actually make a movement. But we had to implant this new area of the brain, very much new territory. And if you look at the posterior parietal cortex, its anatomy in different individuals is very different for each person. So it's a bit like a fingerprint. And it's a very complicated pattern of gyri and sulci, almost like spaghetti, very big noodles. And so it's difficult to know where particular areas are. And we wanted to identify grasp and reach areas that we had found in the animals. So we asked Eric to imagine reaching and to imagine grasping. And we could use functional magnetic resonance imaging to locate the areas of his parietal lobe that were active when he imagined those different movements. So then we were able, with the neurosurgeons, Dr. Liu and Dr. Lee, to implant those sites guided by this MRI data. And it turned out to be very successful, so we were very pleased with those results. We use computer algorithms that decode from the population what the subject is intending to do. And so we can decode what kind of grasp they intend to make or what kind of reaching movement. And then we can take this decoded signal and use it to control the robotic device or a computer screen. It makes the initial intent to make an action. So it's at a higher cognitive level of planning an action. So it's like the motor cortex says, move the arm to the left. This area says, I'd like to grab that cup of water. So we thought that it would be a complementary area that would provide new information that would help to make prosthetic applications more intuitive to the subject and perhaps more versatile to the subject since it's more general in nature. Another thing we found was that if you, there's a grasp area, so it codes the object that is graspable, its shape, and also the shape of the hand to grasp it. And that can be a single neuron, so you can get a whole representation of a grasp, and another neuron nearby will produce a different type of grasp and be sensitive to a different type of object that would require that grasp. So rather than needing many neurons from the motor cortex to form the shape of the grasp, you could do it with individual neurons. So this kind of high-level method may reduce the number of cells that you would need to perform these different tasks. So in motor cortex, the left hemisphere controls the right hand and vice versa. In parietal cortex, each hemisphere controls both limbs. And some cells control the right limb, some the left limb. So the hope is that we could also use that kind of signal. So recording from one hemisphere, we could actually do bimanual tasks. We haven't examined this in detail yet, other than we see both control signals there. But ideally, we'd like to eventually be able to use two robotic limbs to do bimanual actions. I was worried in the beginning that he wouldn't have something to take home to, say, cook a meal or something like that, that it wouldn't be of direct benefit to him. What I hadn't anticipated is that the trial itself, the very process of the trial, is a tremendous benefit psychologically and for personal growth. So it gives him a way of getting out of the house. He has a job He goes to work, he's paid for the trials, he's interacting with a number of people, and so it provides a really rich and exciting environment for him. The really exciting thing with the humans is with the animals we infer what they're doing, but we can't actually ask them. Whereas in the humans we can say, what are you thinking about, what were you planning to do, how were you moving that limb, what kind of imagination were you using. So it's really great to be able to get a conscious report from the subject. It's great to see this technology get translated to people that are paralyzed. In basic research, we work on these issues with the hope that someday they'll help patients, but it is a little distant from the actual clinical application. So in this case in particular, it's such a direct translation. I mean, we learn science in the laboratory, and then we can directly bring it to assist the patients. And moreover, what we're learning too is we learn new science from the patients. So we're learning new things that we didn't know existed in the neural code. And so we can now use that new science that we learn from the human studies to perfect even better prosthetics. In the future, one of the challenges will be to make these these implants wireless, although it's not an easy task because there's so much information that's transmitted. But there are several labs working on this, electrical engineering labs, trying to miniaturize all the electronics and make it wireless so that it could broadcast out of the brain without actually having this percutaneous connector.